My name's Adam Cohen. I'm a professor in the departments of chemistry and physics at Harvard. Today I'm going to talk to you about a huge unsolved problem of how to visualize electrical activity in the brain. Now, take a minute and put your hand on your head and think about what you're touching. Within a centimeter of your fingers, there's a piece of the most mysterious and poorly understood matter in the universe. Here's a picture of your brain. Now, if we zoom in by a factor of about 300, we see that this brain is made up of neurons. There's about 100 billion of them, and they're connected to each other, each by about 10 to the 4, 10,000 connections. If we zoom in again, now by a factor of 10,000, we see that each one of these neurons is encased in a lipid bilayer, a little layer of grease, which separates the inside of the cell from the outside of the cell. This bilayer is an electrical insulator, while the cytoplasm and the extracellular medium are conductors. So you can think of it a little bit like a parallel plate capacitor. Now, there are many proteins that sit in the membrane, and these proteins can pass ions across the membrane. There are charge pumps, which can generate a voltage, and there are ion channels, which can relieve this voltage. These channels act a little bit like resistors, except they're nonlinear. So if there's a change in the voltage, they can have big changes in the conductance, which can lead to these electrical spikes in the membrane voltage, which we call action potentials. Every action potential is about 1/10 of a volt high and lasts about 1/1000th of a second. These are the currency of the nervous system. So every thought and hope and dream and feeling that we have is encoded in these patterns of electrical spikes traveling through the 100 billion neurons in our brain. That's all there is. And so we'd really like to see these action potentials so that we can understand what each neuron is doing, and also to look at the global patterns of activity and the interrelations of the signals between neurons to understand how information is being processed within the nervous system. So basically, this is a voltage measurement problem. Now, traditionally, this is measured as you might measure the voltage in any electrical circuit. Electrophysiologists take a glass pipette with a little electrode in it and jam the pipette into a cell, and then there's another electrode out in the solution. And by hooking these up to an amplifier, they can measure the voltage difference. And this works nicely, but it only measures one cell at a time. It's extremely slow and laborious to get these electrodes into the cell, and it doesn't give you the global view of what's going on in a complex neural circuit. Here's an artist's conception of what we'd really like to do. We'd like to just make these neurons light up when they fire. The problem is, you can't see the voltage in a neuron any more than you can see the voltage in a telephone cord when somebody's having a telephone conversation. We need some contrast agent to convert membrane voltage into light. Now, physicists and chemists and biologists have been working on trying to invent contrast agents since the late 1960s. And this activity has really taken off in the last few years. Here are just a few of the things that people are working on today. These are all from within the last year or so. People are trying to develop voltage-sensitive dyes. So these are organic small molecules which will intercalate into the membrane. And in some of these dyes, illumination with light generates an electronic excited state. The charge distribution in the excited state is different than the charge distribution in the ground state. And so when there's a voltage across the membrane, that voltage perturbs the relative energy of these two states, which can alter the fluorescence properties of the dye. And so this provides a means to read out membrane voltage with fluorescence. Other people are trying to put semiconductor nanocrystals in lipid membranes. These nanocrystals have quantum mechanical delocalized electronic states. Membrane voltage can again shift the relative energies of these states and adjust again the fluorescence properties of these crystals. Other people are working with objects called nitrogen vacancies in diamonds. These are tiny quantum mechanical defects in diamonds, and people think that if you get a small enough diamond nanocrystal and you stick it into a membrane, the fluorescence properties of these nitrogen vacancies will again be sensitive to membrane voltage. Now, I want to emphasize these are all interesting prospective technologies, but none of these has solved the problem that I posed at the beginning yet. In all of these technologies, the sensor is sitting in the membrane. And this is because to measure a voltage difference, you need to be connected both to the inside of the cell and the outside of the cell. Other people, including myself, are working on voltage indicators based on fluorescent transmembrane proteins. The idea is to have a protein which sits in the membrane and to somehow make its fluorescence sensitive to the membrane voltage. I'll tell you just a little bit about one indicator which we've been developing in my lab. The story starts here. This is a picture of the Dead Sea. There's a microorganism that lives in the water here that produces a transmembrane protein called arcuidopsin 3. This protein absorbs sunlight and uses that energy to pump a proton, a charge, from inside the cell to outside the cell. This generates a membrane voltage which this creature uses to power its metabolism. So a few years ago, I said to myself, I wonder if we can run this thing in reverse. Can we use a change in membrane voltage to induce a conformational change in this protein, which will lead to a fluorescent signal that we can see, just like this, right? And you see this works in PowerPoint, and so that's very encouraging. After several years, we engineered a protein which had these attributes. And here's a picture of a human tumor cell where we've introduced a gene for this protein. And we're using a little patch pipette, which you can't see, to induce steps of voltage between plus and minus 1/10 of a volt. And we're looking at the fluorescence of the protein. So here you can see that as the voltage goes up, the protein gets bright. And as the voltage goes down, the protein gets dim. So this is a genetically encoded fluorescent reporter of membrane voltage. And it turns out to be quite fast and sensitive, but also very dim, which makes it hard to measure. We've now developed technology to take these proteins and to express them in neurons in a petri dish. So the neurons become fluorescent, and their fluorescence becomes sensitive to the voltage in the membrane. We're also expressing another protein in these cells, which renders them sensitive to blue light. This is a light-gated ion channel, and targeting a flash of blue light to the cell body will trigger the neuron to fire. We've developed computational techniques to infer the propagation of these electrical impulses at extremely high time resolution. Here I'll show you a movie at 100,000 frames a second of this optically induced action potential propagating through a neuron. So you can really see how these signals propagate through a cell now. But this is a neuron in culture. We've taken the rat brain and we've mushed it up and we've spread out the neurons in a dish. We'd really like to do this in an intact brain, but neither we nor anybody else on the planet knows how to do that right now. There are several challenges to this, and I'll tell you about them. So one of the problems we face is that the brain is not transparent. Here's a picture of a mouse brain, and you can see it scatters light very strongly. It's about the size of my pinky, but light scatters over a distance less than 1/10 of a millimeter. And so it's an unsolved challenge how to visualize what's going on inside of this chunk of tissue. We need to come up with new kinds of optical or electrical tricks. Another challenge is that in every mouse brain, there's about 75 million neurons. To record from these at 1,000 frames a second, which you need to detect action potentials, would lead to a data rate of about 75 gigabytes a second. We don't know how to store this data, let alone what to do with it if we did have it. So this brings me to another unsolved challenge, which is the interpretation problem. So even if we had the best technology imaginable for visualizing electrical activity in the brain, how do we interpret it? We'd like to relate the electrical activity to some statement about the underlying biology of the cells. What are the ion channels that are active? What's their state of regulation? What's their distribution? What's the metabolic state of the cell? What's happening to its energy supply? We all experience that as we go through our day-night cycle, the function of our neurons changes tremendously, whether we're asleep or awake, tired or not tired, hungry or not hungry. And those changes are not mediated by structural changes in the brain, but by changes in, presumably, the metabolic state of our neurons. And then finally, we'd like to infer from patterns of activity how the neurons are connected to each other. Can we figure out who's calling the shots and who's controlling whom, and how they integrate and process information? To illustrate this, here's a movie of some neurons. This was a movie taken in my lab. These are neurons in a dish, not in an intact brain, where we'll look at their firing patterns. This is a movie at about 500 frames a second. The neurons have been artificially colored just so you can tell them apart. And we can zoom in on any one of these cells and look at its firing pattern. And we really don't know how to interpret these firing patterns in terms of the ion channels, the energetics, and the connectivity of these cells. So there's tremendous scope over the coming years to combine the efforts of physicists, chemists, and biologists to develop technology which will let us visualize the activity in this little chunk of matter which is right between all of our ears.